Hello everyone. Our today's session is regarding the laboratory diagnosis of viral diseases, wherein we will cover all the older and the newer methods included in the diagnosis of virus detection. Now we will see which are the various methods available for the viral diagnosis. First is microscopy. Second is detection of viral antigen and antibodies. Third is isolation of the virus. And the last is molecular methods. We shall first see the microscopic methods. First one is electron microscopy, second is fluorescent microscopy, and third is light microscopy. We will see one by one all the three. The use of electron microscopy is quite increased these days, and the viruses are identified on electron microscopy based on their distinct appearance. As for example, the rotavirus. It is wheel shaped. It has got a appearance that is wheel shaped. Then rabies virus. The appearance of rabies virus is bullet shaped. And the very known right now, the coronavirus, the appearance is crown shaped. It has got petal shaped peplomers. Electron microscopy is preferred as a primary tool for diagnosis in situations where the viruses are quite difficult to cultivate. Also, electron microscopy can be used as an alternative to the tissue culture because tissue culture is quite technically demanding. Now, while there are pros, there are cons of electron microscopy, that it is highly expensive and also it has low sensitivity and low specificity. The next is fluorescent microscopy, wherein direct and indirect immunofluorescence techniques are used. As you can see here in the direct immunofluorescence, the specimen is mounted on a slide which is stained with specific antiviral antibody which is tagged with a fluorescent dye and finally viewed under fluorescent microscopy. While in case of indirect immunofluorescence, anti-mouse antibody is tagged with the fluorescent dye against the virus specific antibody. The third one is light microscopy, which you are all used to it. The use of light microscopy in the diagnosis of viral diseases is quite limited to only histopathological staining of tissue sections, where we can see inclusion bodies. Now, what are inclusion bodies? These are virus-specific intracellular globular masses which are produced during the replication of virus inside the host cells, and they are visible under the light microscope. These inclusion bodies could be of two types, intracytoplasmic or intranuclear. Different inclusion bodies are seen in various types of viruses. For example, Negri bodies in case of rabies. and owl eye appearance in case of cytomegalovirus. You can see here the owl eye in cytomegalovirus, which are intranuclear inclusion bodies, and the negri bodies here in case of rabies, which are intracytoplasmic inclusion bodies. The next method is antigen and antibody detection in serum and other samples. Now there are older and newer tests available for the detection of antigen and antibody. The older tests are the hemagglutination, neutralization, complement fixation, and immunoelectrophoresis, and the newer are the immunochromatography, ELISA, and Western blot. Now antibody detection from serum is one of the most commonly used method in diagnostic virology. If you could recall this graph from the chapter of immunity, the primary and secondary response, if you remember this, the primary immune response and the secondary immune response, when the antigen is first exposed, when the first exposure of the antigen occurs, the titer of the antibody is low and it is mainly of IgM type. And when the second exposure occurs of the same antigen, the titer of the antibody is quite high and it is mainly of IgG type. And based on this, various virus infections are detected, whether they are recent virus infections or chronic or past infections. Appearance of IgM antibody or fourfold rise of IgG antibody will indicate recent infection, whereas only presence of IgG antibody will indicate any chronic or past infection. A very commonly used method for antigen and antibody detection is immunochromatography. As you can see here, the classic example of detection of dengue virus infection, IgM, IgG, and the control line, that is the antibody detection, and NS1, the control line, that is the antigen detection. The other method available is always ELISA, that is enzyme linked immunosorbent assay, that you are all aware of, and finally the Western blot test. In animal inoculation, specimens are inoculated into animals like mice or monkey through intracerebral or intraperitoneal routes, and then these animals are observed for any signs of disease or death, and later on these animals are sacrificed and their tissues, they are subjected to the histological examination. Because of the ethical issues related to the animals, the use of animal inoculation is restricted to research purposes, for the study of pathogenesis, for the study of oncogenesis, and for primary isolation of viruses which are difficult to cultivate otherwise, for example arboviruses. The next method of virus isolation is egg inoculation, wherein we use embryonated hen's eggs, and these embryos are hardly of 7 to 12 days. Once the virus is inoculated inside the embryonated egg, the virus produces visible lesions which are called as pox. As you can see here, this is the schematic diagram of an embryonated egg. The samples are inoculated inside this embryonated egg via 4 routes. The first is the chorioallantoic membrane inoculation, wherein the specimen is inoculated via the chorioallantoic membrane. The second is amniotic inoculation, wherein the specimen is inoculated in the amniotic sac. The third is yolk sac inoculation. The specimen is inoculated in the yolk sac. And the third is allantoic inoculation, wherein the specimen is inoculated in the allantoic sac. These are the examples of different viruses which are inoculated via their respective routes. And the last method of virus isolation is the tissue culture. The tissue cultures can be of three types. Organ culture, wherein small bits of specific organs are used to cultivate certain fastidious viruses. For example, tracheal ring culture in case of coronavirus. But this method of organ culture is not used now, it was previously used. The second method is the explant culture, wherein the fragments of minced tissue can be grown as explants. And this method is also almost obsolete now. And the third is the cell culture, which is very routinely employed culture right now. Now we shall see the cell line culture in detail. Now for cell line culture, what we first need to do is preparation of the cell lines. For the preparation of cell line, the tissue is minced into smaller fragments, which is incubated with certain proteolytic enzymes, which will disperse the cell and disassociate the components into the individual cells. Now these cells are transferred in a tissue culture flask, which already has viral growth media inside it. Now on incubation, these cells will adhere to the glass surface and start growing. This will form a monolayer sheet, and this entire process is called as monolayer sheet formation of the cells. These tissue culture flasks are then incubated horizontally. As you can see, the virus will then start using the cells and grow themselves. Now these cell lines are classified into three types based on their origin and maximum number of cell divisions that they can undergo. The first is primary cell line, second is secondary and continuous cell line. The primary cell lines, they are derived from the normal cells which are freshly taken from the organs. They are able to undergo 5 to 10 divisions. The example is monkey kidney cell line, chick embryo cell line, etc. The secondary cell line are also derived from the normal host cells, and they maintain the diploid karyosome. They are able to undergo 10 to 15 divisions. Examples are human embryonic kidney and skin fibroblast cell line. And the third is the continuous cell line. The continuous cell lines, they are derived from the cancer cell lines, and hence they are capable of indefinite growth. They are able to continuously divide. The example are HeLa cell line from human carcinoma of cervix, Hep2 cell line, human epithelioma of larynx cell line. Now how do we detect the growth of the virus in these cell cultures? So there are various methods available for the detection. The first is the cytopathic effect, in which basically the morphological change produced by the virus in the cell line are detected by the light microscope. And these cytopathic effects are cell degeneration, syncytium formation, cell rounding or ballooning, and cellular clumping. There are very few viruses which can produce cytopathic effect, and these viruses are called as cytopathic viruses. The second method of detection is viral interference. The growth of a non-cytopathic virus in cell culture can be detected by the subsequent challenge of the cell line with a known cytopathic virus. Here, the growth of the first virus will inhibit the infection by the second virus, that is the cytopathic virus, by a mechanism which is called as viral interference. And the other methods are hemadsorption, immunostaining, immunofluorescence, electron microscopy, and gene detection. You can see the examples of the cytopathic effect: the syncytium formation in case of measles virus, heme adsorption in case of mumps virus, and ballooning of the cells in case of herpes simplex. And the last group of methods which has made the diagnosis of virus disease very simple and smoother are the molecular methods, that is the viral genome detection. There are two methods, that is hybridization, and the very known that you are all aware of is the PCR, that is polymerase chain reaction, and the types that are the RT-PCR, reverse transcriptase PCR, and real-time PCR. Now the conventional PCR involves three basic steps: the DNA or RNA extraction, DNA amplification, and the detection of the amplified products by gel electrophoresis. As you are all aware of the test that is being used right now, that is the real-time RT-PCR, the real-time reverse transcriptase PCR, which is used for the detection of RNA viruses, I'll just go through superficially through this test as it is the word of the town right now. So the sample in the COVID-19 molecular diagnostic test through RT-PCR is the nasopharyngeal or the oropharyngeal swab, which is collected in a viral transport media. This viral transport media is then brought into the sample processing room, wherein we basically deactivate the virus. Then the next process is the RNA extraction. The RNA is extracted from the deactivated virus. Then this purified RNA is reverse transcribed to the complementary DNA and amplified using PCR, that is the thermal cycler. And then the test results are as shown in this picture. They are depicted in the form of the graphs. So the interpretation is basically done based on these graphs. The last one is the viral assay. These assays are basically used for the quantification of the virus particles. They can be grouped into physical assay and biological assay. The physical methods includes the real-time PCR, antigen detection via ELISA, hemagglutination assay, and electron microscopy. And the biological methods, both qualitative as well as quantitative methods are available. In qualitative, endpoint assay, and in quantitative, Plaque assay or the POC assay. Plaque assay is the most widely used assay for the quantification of infectious viruses. In this, the monolayer of the cell line is inoculated with suitable dilutions of the viruses. Multiple cycles of the replication and cell killing will produce a small area of infection, which is called as the plaque. As the single plaque arises from a single infectious virus particle, the number of plaques will represent the quantitative infectivity titer of the virus suspension. As you can see here, the number of plaques. I would like to end the session on a happy note that the real-time RT-PCR test that we just discussed is now being performed at the molecular laboratory, Siyusha Medical College, and the pictures here are the real pictures of our lab. Thank you.